So, hello everyone. To start with the next video, that is on PCR components. So, we know that PCR is the amplification of single DNA molecule into multiple or millions of copies of DNA strands, right? So, some components are used to amplify these DNA molecules. So, the component of PCR components are, one is polymerase buffer, next one is dntps, and your polymerase. Then is your forward and reverse primer, then your water, and then your magnesium chloride. So we'll discuss all these components one by one. Primers, forward and reverse primer we have discussed in previous lectures. So now we'll discuss polymerase buffer. Why it is known as polymerase buffer? Because what is a buffer? Buffer is a system which prevent the change in the pH, right? So polymerase buffer means the buffer required for the polymerase activity. We know that for the amplification of PCR, we need or we use DNA dependent DNA polymerases, right? For those DNA dependent DNA polymerases, we need a buffer system. So, buffer system, a buffer which prevent the change in the pH. So, for the activity of polymerase enzyme, we need some components like trace Cl. What is trace Cl? Trace is a base whose pH is more than 10.5 or close to 10.5 or 11, and whose pH is also adjusted with HCl and brought down to pH 8 to 9 point, between 8 to 9.5. And at this pH, this TricCl it will act as a buffer system. It will prevent the change in the pH during PCR reaction. So along with TricCl, there is another component, that is potassium chloride. What potassium chloride, what it will do? It will increase the binding of primers to the template. How it will enhance the binding? Because it will provide positive charge potassium ion and negative charge chloride ions. And potassium, these ions, it will surround your template and primer duplex. It will surround potassium ion like that, and potassium ion like that. Because outside the duplex there is negative charge, so it will attract all the positive charges. So it will enhance the binding of primer with the template. In some polymerases which are having high fidelity, they have exonuclease activity. So those have, along with potassium chloride, these buffers, they have ammonium sulfate. So what it will do? It will also ionize to ammonium ion and sulfate ions. So these ammonium ions, it will also surround the duplex. But what it will do, because it has sulfate ion also, it will prevent the binding of primer to some non-specific site. For example, if this is your template 5' to 3', and primer is binding here, but it has only 4 complementary bases instead of 20 bases or 25 bases. So these 4 complementary bases, because if it will bind and it will extend, it can give non-specific bond. So to prevent that, the ammonium ion, it will surround this duplexes, and also the sulfate ions and the sulfate ions also prevent or it will melt the formation of non-specific primer and template complex. So like that, these two buffers, they have only one target. Target is to amplify the DNA into millions of copies. So potassium chloride will enhance the binding of primers to the template, and ammonium sulfate, it will prevent the binding of primer to the non-specific site. So like that, because these two components are the part of buffer system of exonucleases such as Wnt polymerase or PFU, like that, or fusion enzyme, those having high fidelity property, exonuclease activity. So these two buffers are, these two molecules are used. So, but here one thing you should note, that when the buffer system is only using potassium chloride, so I will write again, if polymerase buffer is using only potassium chloride, then the magnesium chloride concentration should be 1.5 millimolar, or with plus minus 0.2 millimolar. But it should not be more than 1.5 millimolar. Otherwise, you will get non-specific amplification. But when the exonuclease enzymes were used, in those buffers, ammonium sulfate is present, and for ammonium sulfate containing buffer, the magnesium chloride concentration should be high. So it is around 2 millimolar plus minus 0.2 millimolar. A higher concentration of magnesium chloride is used when they are using ammonium sulfate containing buffers for high fidelity enzymes. So that part we have to take care. If magnesium chloride concentration is already present along with the buffer, then we don't have to adjust or we don't have to standardize. But otherwise, for some PCR or for some reactions, we have to standardize the magnesium chloride concentration along with when we are using high fidelity enzymes. So another question is, when we are using some GC-rich template like rubella genome, where the maximum G's and C's are present, because G and C, they are linked with triple bond, so it is very difficult to melt those bonds. So GC-rich template, for the GC-rich template, we use some, in addition to the buffer, we use to melt the template, we use DMSO around concentration 1 to 10%. Then we can use betaine 1 to 3 molar. Then we can use formamide 1% to 10%. Then we can use BSA, bovine serum albumin, and that is 10 to 100 microgram per ml. Then we can use Triton, which is also part of your Triton or Tween. These are detergents. And then we can use glycerol. And for whenever we want to use these components, we have to standardize according to our PCR in whatever we want to amplify in our lab. So all these components we can add to GC rich containing templates. Wherever we are working with GC rich, that time we have to use, or we have to select one of them to melt the template, so that if a successful PCR can be obtained. So with this is all about polymerase buffer and the components of the polymerase buffer, which are DNA dependent DNA polymerase, and the type of buffers we are using, either for your Taq polymerase, which is which do not have exonuclease activity, and and the buffer for the exonuclease activity, enzymes having exonuclease activity. So thank you for all, and next video will study the dideoxynucleoside triphosphatases, that is DNTPs, another component of PCR reaction. Thank you.